Good day. In this lecture, we will be discussing about the cytoplasmic membrane systems. So for a brief layout of the lecture, we will first look at an overview of what you call the endomembrane system, which are composed of the ER, endoplasmic reticulum, the Golgi body, and then we'll talk about vesicular transport and post-translational uptake of proteins. So first, let's look at the overview of the endomembrane system. But first, what is an endomembrane system? So the endomembrane system basically describes the organelles, which is, by definition, they are the organelles that are part of a dynamic integrated network in which materials are shuttled back and forth from one part of the cell to another. Now, let's look back at the definition one by one. It's a dynamic network, meaning materials are constantly being synthesized or degraded. The endomembrane system are constantly changing. And it's also an integrated network. They are connected with one another. And one of the main functions of the endomembrane system is to shuttle materials. So what are those materials? They can be protein, newly synthesized proteins. They can be cellular products. They can be hormones. They can be molecules, signaling molecules. They can also be debris from bacteria and viruses. They are being shuttled back and forth. So materials are shuttled between organelles, basically from the Golgi complex to the plasma membrane, for example, in small membrane-bounded transport vesicles that bud from the donor membrane compartment. So these transport vesicles move through the cytoplasm in a directed manner. They are often pulled by motor proteins that operate on tracks formed by microtubules and microfilaments on the cytoskeleton. So when it reaches this destination, the vesicle fuses with the membrane of the acceptor compartment, which receives the vesicle's soluble cargo as well as its membranous wrapper. So here is a transmission electron micrograph of your several parts of the endomembrane system. So let's look at this one fully. So we have here the Golgi complex, the one that looks like a bunch of cotton buds. And then you have the endoplasmic reticulum. And then these small clear vesicles at the bottom are the secretory vesicles. So the shuttling, basically the vesicular transport goes something like this. So you have this material that needs to be shuttled back and forth. Basically, you can look at like packages that you ordered from either Shopee or Lazada. So you have your packages, the soluble proteins that are this comes from we are going from the bottom up, the bottom up movement. So we have these protons from the donor, the initial compartment, the donor compartment, or the source, basically. So they are packaged. When we say package, they are selectively packaged on a particular vesicle. So you have this invagination of this compartment. They form vesicles, and this vesicles travels to the recipient compartment. When it reaches that one, since this is a membrane, so it's the same consistency as our cell membranes. It's made up of a lipid bilayer, so it's easy to segregate and it's easy to actually fuse. So they just simply fuse to the recipient, and then you get the recipient donor. So basically, that's the vesicular transport. And this vesicular transport forms part of your biosynthetic pathway, also called your secretory pathway. So it is the route where proteins are synthesized in the endoplasmic reticulum, modified during passage through the Golgi complex, and transported from the Golgi complex to various destinations. So we have two types, basically, of secretions. We have the constitutive and the regulated. In the constitutive secretion, materials are transported in secretory vesicles from their sites of synthesis and discharged into the extracellular space in a continual manner. So it's called constitutive because It's always on, it's always there, it's always happening. Whereas in the regulated secretions, the materials are first stored as membrane-bound packages and discharged only in response to an appropriate stimulus. So it's called regulated because the discharge, the use or the discharge of this secretion is highly regulated, only when needed. They are only discharged only when needed. So here is another view of this secretory pathway. So let's start with the initial synthesis of them. Basically, most of these materials are basically protein-based materials, protein sources. They are enzymes. Most of them, they can be enzymes, they can be hormones, regulatory hormones. So the synthesis, let's say for example, if the secretion or the material is a protein, exogenous protein. So let's have this the first, of course, the synthesis of any proteins usually occurs via the central dogma. So you have the transcription of the gene via mRNA, and then translation of the mRNA to the proteins, and that happens in the rough endoplasmic reticulum. So the materials are in the rough endoplasmic reticulum. So it travels via vesicular transport to the Golgi complex, and then in the Golgi complex, so this is from the previous image, this is a more expanded view of that bunch of cotton buds. So it's basically a layered like structure. So you have it's quite distinctive when you look at it in real life in the microscope. It's quite distinctive. It forms this type of layers, several layers. So there is a travel. The material travels across those layers, and then finally to their appropriate final destinations. So if you have your constitutive secretory pathway, it's always on. That means it's continuously being secreted upwards. If this is to be an exogenous protein, it's supposed to go out to the cell. If it's regulated, unless it's needed, it stays hanging around the inside of the cell. Only when a certain stimulus appears, that is the only time when it will be released outside. And then the same goes for the other side. So for an endocytotic pathway, that is your cells invaginates or takes up materials from the outside. Usually, for example, macrophages eating up bacteria and other cellular debris. So they do phagocytosis or even an endocytotic route. So this when our cells invaginate or gobbles up this material, they form your endosomes, basically a vesicle inside. So the materials from the outside are trapped inside this endosome. And these endosomes are being fused with materials from the inside. Usually, these are materials that contain digestive enzymes to digest the materials from the outside. And they fuse together, the materials from the outside and the digestive enzymes from the inside fuse together to form your basically lysosome. And that is where your cells digest materials from the outside. So that's the general overview of the secretory pathway. So here is, if this is your artist sketch, this is the actual transmission electron microscope image. So this is an electron micrograph of a section of a pancreatic acinar cell that's been incubated for 3 minutes in radioactive amino acids and immediately fixed and prepared for autoradiography. So you have the radioactive amino acids so that you can easily visualize what happens to that amino acids. So the black silver grains in the that appear in the emulsion following the development are localized over the endoplasmic reticulum. So basically, the dark grains are your radioactive particles. So you tag them with radioactive particles in order to view what happens to them. So in the regulated secretions, materials to be secreted are stored in this large, densely packed membrane secretory organelles. So these are the dark ones. These are the secretory granules, and these are due for regulatory secretion. So basically, they are large, but they are hanging around inside the cell. So they're just packed inside. Whereas the smaller ones, So you have smaller silver grains of other, because it's basically just amino acid. So it can be a regulated protein or a constitutive protein. So others are basically used by the cells. So they form these small silver grains, so hanging around in other parts of the cell. So these are mostly for constitutive tasks. So here is a diagram of the sequence of the autoradiograph showing movement of the labeled secretory proteins. So from continuing on with that experiments, here is the image of that one. So basically, you have the secretory granules, the light blue ones, that are hanging around. So after being produced or synthesized, the proteins synthesized by the cell using those radio-labeled amino acids. So they form the secretory granules. They hang around just near the site where they would be excreted in case that they need to be excreted. And then, so after 20 minutes, so you have your um, secretory granules. So this granules becomes concentrated in the secretory granules, and then they are released outside. So basically, what does this diagram shows us? So that means that the cells, when they synthesize these proteins from the radio-labeled amino acids, so basically not only radio-labeled, synthesis proteins per se that are for supposedly for secretions, they are stored first in this secretory granules. So the pathway is basically the RF ER where they are being synthesized, followed by packaging and transport via the Golgi apparatus, and then hanging out around the secretory granules before being released to the outside. So basically, that's the pathway where the secretory granules occur. Now, let's have a brief session break and a recess. So I'll just be finishing this part of the lecture for now, but I'll be giving you the session break questions in the discussions. So I will put up a discussion board and please see the lectures in there. So this is also a graded discussion. So I want to know your thoughts on the following. You can consult journals, you can consult reference books from there, but do not copy directly from ChatGPT. So the questions are, how do each protein and vesicle know which way to go? And in the regulatory secretion, what controls when the vesicles and its cargo is to be released? So both questions, you should be answering that. And additional one for the homework, this is a by group homework, is to look into how the following techniques advance our knowledge on cellular processes. So all of the lectures that we have discussed are actually the fruits of the labor of the scientists using these techniques, but not limited to these techniques, but these are just some examples. So you have autoradiography, fluorescence protein tagging, subcellular fractionation, cell-free systems, and mutant phenotypes. I just want you to create a flowchart on these techniques. So that ends our first part of the lecture.